Very good morning to all of you. Uh, we are going to continue today the part that we have left in the last lecture, that is the citric acid cycle. In the last lecture, we have seen the three first initial steps of the citric acid cycle, wherein there was a conversion from acetyl coenzyme from the 6 carbon molecules to 5 carbon molecules for isocytrate and formation of alpha glutarate that we have seen. We are coming with the same topic, or we'll continue with the same topic. So this was the reaction that we were discussing in the last lecture: isocitrate, oxaloacetate, and alpha ketoglutarate. You can see oxalosuccinate, which is an intermediate product that is being generated when isocitrate gets converted to alpha ketoglutarate. The respiratory chain linked oxidation of isocitrate proceeds almost completely through NADP dependent enzyme. So a particular enzyme is there which converts isocitrate into alpha ketoglutarate. Now, step 4, formation of succinyl coenzyme. Alpha ketoglutarate undergoes oxidative decarboxylation in a reaction catalyzed by a multi-enzyme complex similar to that involved in oxidative decarboxylation of pyruvate. The alpha ketoglutarate dehydrogenase complex requires the same cofactors as pyruvate dehydrogenase complex like thymine diphosphate, lipolate, NAD plus, FEDN coenzyme A, and result in the formation of succinyl coenzyme A. So in the fourth step, the alpha Kg is now converted into succinyl coenzyme A. See here, alpha Kg with the help of NAD and CO, the CO molecule or the coenzyme A has been attached to succinyl coenzyme A. This is an oxidative decarboxylation type of reaction. This equilibrium of the reaction is much favor of succinyl coenzyme formation, that it must be considered by physiological or unidirectional. As in the case of pyruvate deoxidations, arsenate inhibits in the reaction, causing the substrate alpha Kg to accumulate. Fifth step, formation of succinate. Succinyl coenzyme A in an energy-rich thioester compound. The cleavage of thioester bond of succinyl coenzyme A coupled to phosphorylation of purine nucleoside diphosphate, usually GDP. This reaction is catalyzed by succinyl coenzyme synthetase. The GTP formation is used for the decarboxylation of oxaloacetate to phosphoenol pyruvate in gluconeogenesis and provides a regulatory link between citric acid cycle activity and the withdrawal of oxaloacetate from gluconeogenesis. Here, a succinyl coenzyme with the help of one GTP molecule, one GTP again and high energy rich compound is being generated here, and formation of succinate takes place. This is the only example in citric acid cycle of substrate level phosphorylation. This is most important part because it is usually asked in the examination that which is the step which is the substrate level phosphorylation. This is the substrate level phosphorylation that takes place, formation of succinate by succinyl coenzyme A. Next step, formation of fumarate. The first dehydrogenation reaction forming of fumarate is catalyzed by succinate dehydrogenase, which is bound in the inner surface of the inner mitochondrial membrane. The enzyme contains FAD and iron sulfur, FES protein, and directly reduces ubiquinone in the electron transport chain. See here, FAD to succinate, FADH2, the high rich compound has been generated and fumarate has been generated. Next, step 7, formation of malate. Fumarates, that is fumarate hydratase, catalyzes the addition of water across the double membrane bound to fumarate yielding malate. See here, structurally, if you see, this is fumarate, the H2 molecule gets bind, and ultimately, because of this, the structural differentiation takes place, and L-malate has been, or the malate has been generated. Regeneration of oxaloacetate. Malate is converted to oxaloacetate by malate dehydrogenase reaction requiring NAD+. Although the equilibrium of this reaction is strongly favors malate, the net flux in the oxaloacetate because of the continual removal of oxaloacetate to form citrate as a substrate for gluconeogenesis or undergo transamination to aspartate and also the continual reoxidation of NAD. See, succinate to fumarate, fumarate to malate, and malate again to oxaloacetate. So energy yielding acetyl coenzyme turns per cycle. So ultimately, generation of ATP that takes place. We have already seen that the 11 ATP molecules have been generated with this. So one acetate unit generates approximately 12 molecules of ATP. Two molecules of ATP are generated, four molecules of glucose, which generates two molecules of acetylene and anaerobic glycolysis. Molecular oxidation does not participate directly in the citric acid cycle. So yield as comparatively is said to be quite high in citric acid cycle. Regulation. Excess of ATP deficient energy rich state of the cell. Hence the TCA cycle is inhibited while the reverse reaction when the cell is allow energy state with excess of ATP. See here. These are the points by which the inhibition of the cycle would take place and there will not be formation of ATP molecules. Correct. So that's all for today. And we have seen the part of citric acid cycle, which is a more important cycle associated with the metabolism of glucose.